entrepreneur, other, and this is the last one, and this is the last one on the industrial side. It's really a great pleasure, it's a tremendous pleasure to welcome back to campus and to Chemical Institute, Dr. Bill Young. Let me give you a brief introduction about him. He received his bachelor's degree here in chemical engineering in 1966. And in fact, in doing that, he was a classmate of Professor Phil Wonkat. So, Professor Wonkat and he were classmates here, along with my time, and we were back another time for ExxonMobil. Anyway, so then soon, immediately after receiving his bachelor's degree here, he joined Eli Lilly and remained with Eli Lilly for 14 years in Puerto Rico. During that period, learning about the manufacturing practices in the pharmaceutical industry. And during that period, he also received a master's in business administration from University. Then this was until 1980. So 14 years. So 1980, as we now go back 30-some years, that was the time when the biotech industry was just in its infancy, and was just coming up. In fact, Bill Young is one of the founders of the biotech industry of the world, because in 1980 that he joined as one of the very early employees Genentech. All of Genentech as one time the largest biotech company in the world. He remained with Genentech until 1999, 19 years, during which, towards the latter part of that period, he was the chief operating officer for the corporation, Genentech, chief operating officer. Then in 1999, he got to the NC. And after having served and grown this company from nothing to the largest biotech company in the world of its time. Well, he said he needs to start something new at that time. So in 1999, he started a company that he founded called Monogram Biosciences, that then became the leader. It had 400 employees at the end, and he sold it at $90,000. So that went from 1999 to 2009, for 10 years. He was the CEO and was the founder of Monogram Biosciences, this leader in personalized medicine. And then it was time to move on yet again. And in 2010, he joined this current position, which is Clarus company, as a venture partner in Clarus. In addition, he's doing other things too, because one thing is not sufficient for him, he's doing more things. And he's also serving as chairman of the board of directors of Biotech, and is also member of the board of directors of several corporations in addition. Cannot be maintained. He has received numerous recognitions, of course, among the management person of his caliber and his position and the impact that he's had on the field. And one of them is the honorary doctorate in engineering from Purdue in the year 2000. And finally, in 1993, he was elected to the National Academy of Engineering for his leadership in research, development, and manufacturing of recombinant proteins using recombinant technology. So we are extremely pleased and thrilled to have you visit here and to spend a day with our faculty students, and really looking forward to your talk on the history and promise of biotechnology.
Great. Thanks very much. Thank you. So one thing hasn't changed since I was a student here. Everybody's in the back of the room. So thanks very much for that kind introduction, and it has been a busy life. I know a lot of a lot of the questions today were around career and what to do and so forth. So I offer myself after this or any other time you have an interest in finding out more about the industry and where you might fit into it. I'm happy to provide that. I'm gonna sort of set the stage a little bit because in life sciences, which is the field that I love to play in and many of you are already working in, there's a lot going on right now, and a lot of it is still positive. There's a lot of room for innovation, but some of it is I would say, seeding some change that's going to be fundamental. And so I want to take you through that, what's happening there, why it's happening, and what the future looks like. And then I'm going to take you back a few years, back to the late 70s and 80s at Genentech, and talk a little bit about some of the lessons that we learned as we developed the first recombinant products. A lot of the technologies we take for granted now didn't exist then, and so to me, and there was a different set of problems for each of the projects that we took on. To me, there's some lessons there, and I just want to take you through that so you appreciate a lot of the things that we now use and take for granted. I'm then also going to talk about, probably briefly, two companies, but a little more extensively, two other companies that I'm involved with that represent two ends of the spectrum. One, a large, successful biotech company that has a lot of products on the market, and another company that I think has a great technology focusing on personalized medicine. And I'm going to talk a little bit about that as a part of that company, because I think that's one of the next sea changes coming in healthcare. So that's the plan. Hopefully I'll get through it all. But so, life sciences is a huge market. It's a $1.25 trillion market. Much of it is in biopharmaceuticals, about 800 million of that, or 800 billion, you can see medical devices represent about 340 million, and the others are smaller. It's driven by scientific advances that we see every day, by the fact that patients are much more informed now about their disease. If you have a serious disease, you do a lot of research most of the time, much of it available on the internet, to find what your treatment options are. So it's not going into the doctor and he telling you what to do. The patients come with a lot of information. That's changing the way these diseases get treated. There's a lot of work now by the big drug companies, particularly in emerging markets. A lot of potential there, and attention is turning to those. We're all getting older. You guys aren't, but I can tell you I am. And there's going to be diseases that are in larger numbers that we'll need to deal with as the population ages. And we're all not the best at taking care of ourselves, so there are unhealthy lifestyles that we need to overcome by preventative techniques in healthcare. But this is a really vibrant market. The United States leads the world in technologies in this area, and I hope that'll continue for some time to come. If we now dial down a little bit to biopharmaceuticals, we do see some structural problems that are going on that you may have read about. First of all, many of the major drugs developed in the 80s and 90s are coming off patent. And the major drug companies are going to lose over $100 billion in sales in the next few years. And that's a huge hole for them to fill. There are also pricing pressures. The United States still has the highest prices for drugs in the world. In a way, we are subsidizing the rest of the world's drug development. And so that pressure is continuing to increase here, pushing down on prices. I've never seen FDA as conservative as they are now, and I've been watching them for a long time. The pendulum on how much data you need to demonstrate your product is efficacious and safe is way over on the thorough side, driving up tremendously the cost of doing phase 3s and clinical development, and providing a lot more uncertainty. So those are some pretty important changes going on in the industry. If you think about, here's some data that shows that R&D investment is going up, and yet the number of new chemical entities approved by FDA is going down. So this is a reflection of some of the pressures of more complexity in working through the issues around clinical development. And innovation doesn't really scale. The R&D hurdles are higher Now we have the Center of Medicine, Medicare, also opining on whether a drug is worth paying for or not. And as I say, FDA in 2011 only approved 17 new chemical entities. And so, what's going to happen in that equation? We need, it's going to stifle the whole pipeline of activity in innovative drugs. We also have something called the healthcare reform, or Obamacare, that a lot of people talk about. That's passed, but doesn't really become effective till 2014. And, in the U.S., our healthcare costs are rising so rapidly that people are predicting that in 2084 it'll be equal to the GDP of the country. And obviously that can't happen because we have to do a few other things besides healthcare. So how to control that, where the efficiency is going to come from. The new healthcare laws don't really go into effect for a couple more years. There are elections before that. So there's uncertainty in the early stage. In later stages, the outcomes are clear. You're going to need outcomes data to get payment, not just approval. There are going to be more generic products, and there's a whole new class of products called biosimilars, I'll talk about later, which are biochemical, biotech copies of the existing pioneer drugs. As those drugs come off patent, a lot of issues around what really is a biosimilar and how equivalent it is to the pioneer drug, pricing pressures, and I think the result is going to be fewer innovative drugs. The big drug companies are responding to this in a couple of ways. In the 80s and 90s, they were busily developing drugs. They had blockbusters they were selling in the EU, US, and Japan primarily. They had big sales forces. They did all the R&D internally. Then in the 2000s, things are getting more specialized. Some R&D gets externalized to biotech companies. And I think what's going to happen now going forward is that's going to be even more done. And you've seen drug companies laying off their sales forces. That happened a few years ago. Now it's R&D. So Pfizer is cutting their R&D budget from $12 billion to $6 billion over the course of a couple of years. That's a lot of R&D to go away, and something's got to fill the innovation for that. Also, drug companies are much more interested in the developing world, so there's a lot more effort being spent selling products in China and India and those places. Now the downside is the margins are much less there. So you can get revenues, you can't necessarily get profits. But anyway, there's this whole sea change. Now some drug companies are reacting by diversifying further, but others, Abbott would be a good example, just in the news the other day, is doing just the opposite. They're dividing into two companies, a drug company and then an everything else company. And, there are going to be two public companies and they'll be judged on their own merits. So the whole point here is that lots of things happening, a lot of dynamics in the industry. Nobody's quite certain where it's all going to come out. There's still, these are all public companies. There's still a lot of demand for growth. And so how do you do that? Well, as I said, you reduce R&D, you reduce operating costs, you merge so that Wyeth is bought by Pfizer, and then one whole layer of management goes because you don't need two of everything. So that's one way to do it. You can reduce internal R&D and outsource to biotech. That's now happening. You can see that. And then the deals that you do are de-risked. That is, you're putting the burden on the smaller company with benchmarks and milestones as the drug progresses through the clinic. And so if it doesn't work, you're not out a huge amount of money. And then diversification is happening. To grow the top line, you can just buy sales. Enter emerging markets. As I say, you get more sales, you don't get necessarily more profits. Buy products from biotech companies. There's also a trend, and one of my companies does this too, to buy companies that are focused on orphan diseases. So these are often genetic defects. They're enzymes that are lacking in certain patients. There might be only 2,000 or 3,000 of these in the world, but you can really help those patients by developing drugs just in that space, and it's limited in clinical trials and development costs, and you know what you're doing. So there's Genzyme was just acquired by Sanofi, and that's largely their business model. I think the big drug companies are doing what maybe they should have been doing all along. They're kind of morphing into biotech. So Roche has acquired Genentech. Now they have a big biotech presence. BMS bought Zymogenetics and Metarex. AstraZeneca bought MedImmune, so they're kind of acquiring the assets and people and skills to be biotech companies also. This is just a depiction of the patent cliff to put it on a little more perspective for you, and you can see at the top of the graph are companies who are going to lose major sales. Actually, one down the road in Indianapolis is one of the biggest, has one of the biggest set of problems around this. It's projected they'll lose 10 to 15% of their sales as patents go off on some of their key drugs. Others are better positioned. They've been acquiring products, they have pipelines, and I think, but again, this is the way the industry is looking today. There also is concern on the part of public investors that R&D internally has actually wasted money. You'd be better off not doing the R&D, buying the product from a smaller company, higher efficiency in the use of capital, and you could give some of that money back to the shareholders. And I think part of the reason is the fixed costs in R&D and pharma are very high relative to biotech. So that's an opportunity for us. I won't dwell on this, but it just shows that there's a move on pharma's part to buy later stage products rather than earlier stage products. So this is also affecting my industry, the venture capital industry, because the partners are looking for things that are further developed with less risk. It means we're starting as venture capitalists fewer new companies. So now I tell my more experienced colleagues or ask them, aren't you worried in 5 years there's not going to be much of a pipeline of new companies and new product ideas in order to keep funding the industry? They say, no, this is just a cycle, it'll reverse back when people realize this is happening, but I'm not so sure. Because I know in our firm, we're only looking at later stage assets to fund and things that have a 2 or 3 or 4 year inflection point, not technology platforms that have just started or are going to take 10 years to develop. It used to be, back when we were all at Genentech, that IPOs and initial public offerings were the absolute best way to monetize the company, give shareholders money back, the investors that invested in employees, and the IPO market is pretty well dead. You can still see them for internet companies, but for healthcare companies, they're rare and far between, and when they are done, there's a significant amount of insider participation. That is, the existing investors also invest a lot in the IPO. So it's not really a liquidation event for them. It's a way of getting access to the public markets and getting more money, but it doesn't really help you with the endpoint game. So, and I don't see that getting better. And there are a lot of reasons why the IPO market is stuck, but I don't see that improving in the next few years. I'll come back to this in a bit, but I think one of the things that one segment of the healthcare market is diagnostics and tools, and one of the reasons that I think this is exciting is that some of you remember Moore's Law, which is the cost of computing drops an order of magnitude every 10 years. I think that's kind of what he said, and that's been the case. It's certainly going faster in the cost of DNA sequencing, and now it's predicted that you could get your whole sequence for $1,000 in the not too distant future. There's next generation sequencing, so the amount of information being generated by genetics is astounding. There's issues about what to do with all that data and how to turn it into useful information. But I think this can be a strong market and a very interesting one, and plays into personalized medicine a little bit later. Question will be, with all the federal deficit issues, with issues around funding of NIH and others where a lot of the money comes from, what's going to happen there? Is that going to dry up? We're already seeing in NanoString's customers, this little company in Seattle, people holding back, waiting to see what's going to happen before they buy the next $250,000 instrument. So this is a question, but hopefully it's a temporary one, and we'll get over this and keep getting good financing of basic research out of the federal government, which I think is their role. So let me now I'm going to go back a little bit further and talk a little bit about Genentech and a few of the lessons learned. I'm not going to be highly technical here, but you might be interested in some of this. I think probably we need to write a book before all of us pass away out of the industry, but if you take yourself back to 1976 when Genentech was founded, this was a company founded by Swanson, who was a venture capitalist, but actually was looking for something to start, and he was in the Bay Area, MIT grad, chemistry, and Herb Boyer, who was a professor at UCSF in the Bay Area. And Herb spent his whole career studying restriction enzymes. And so that was kind of his thing, and he developed a number of these enzymes and characterized them. And then he ran into Stan Cohen at Stanford, and he had spent a lot of his career working with plasmids. So these are little circular pieces of DNA that shuffle in and out of bacteria. And they got together one day, probably over a beer, and said, hey, take your restriction enzymes and your plasmid technology, maybe we could express a human protein here. And that really became the foundation principle. And this was in the 70s, this was early 70s. And that became kind of the foundation idea of Genentech. So Bob and Herb got together. Bob had talked to every scientist in the field, I'm sure, including Cohen. Everybody told him, no kid, this is years and years away, you'll never do this. But in Boyer, he found a willing partner. And so the two of them formed a company. They each put in $500 and started Genentech, and then tried to figure out, okay, now what do we do? They moved to South San Francisco in 1978, which is south of the airport, if you've been to the Bay Area. Actually, it's north of the airport, but is kind of an industrial area, and then was steel plants, fabrication plants, meat packing, but a lot of freight forwarders. And a lot of the first two industries had closed up and moved out. So he was looking for a place that would be very pro-industry. At the same time, in Cambridge and Berkeley, you had Cetus and Biogen trying to clone human insulin, and both towns banned all genetic engineering. Remember, you guys don't remember, but there was a concern about, will this actually be harmful if we start manipulating these organisms? And there's a whole regulation set around that. But both those towns stopped the research. But South San Francisco didn't have quite the same level of concern. They were interested in business. So we located there anyway. And the first commercial agreement that Genentech did in '78 was on human insulin. Lilly was, at the time, the biggest producer of insulin. Most of it was made from the pancreas of cattle and pigs. They could see the curves where the incidence of diabetes was going like this, and the availability of animal pancreases was flat, and they could predict they were going to run out if they didn't come up with a better way of producing human insulin. That was their motivation. It was also clear that the animal origins were a problem. You couldn't totally purify all those out. So there were two reasons why Lilly was very interested. And so Lilly and Genentech did this agreement, and that was the very first genetically engineered product. And I'll talk a little bit more about some of the issues around that. This is a little immodest, but I did move to Genentech from Lilly in 1980. So I worked on the human insulin project and then saw it from the other side. Genentech did go public in 1980. This was much like if Facebook went public, very big news. Then it was in the New York Times and Wall Street Journal. The stock went out at 35, went up to 100. Everybody was very excited. It was one of the first biotech company, or the first biotech company to go public. Believe it or not, we did get insulin approved in 1982. That's pretty fast. You talk about a 5-year project to get a new drug approved. That can't be done today. But several of Genentech's products actually did hit that. Growth hormone, which I'll talk about, was approved in '85. Then a product for heart attacks, Activase, was approved in '87. Pulmozyme, I'll talk about all of this. And then we got into monoclonal antibodies. I remember a lot of debates we would have in the company. Are proteins dead? Gee, we've kind of gone through all the useful therapeutic proteins. And then somebody came up with the technology to humanize monoclonals, to target them, and to make them at scale, and suddenly you had an unlimited number of potential products. Sadly, Roche acquired all of Genentech that it didn't already own in 2009, so now it's a division of Roche, and many people have gone on to bigger and better things. But, very important bellwether company. I got to Genentech in 1980, and they said, would you mind setting up a process science and manufacturing group? I said, well, is there something here to start with? They said, no. So in all of the work around insulin, that was done by Lilly. I like this quote here because mountains cannot be surmounted except by winding paths, and that's been the experience in this technology development exercise. You need to make some false starts and go solve the problems and then move forward, and you learn a lot from that. There were a lot of people at Genentech that did this work. I didn't. I led it, but at one point we had 250 scientists in process science at Genentech, spread amongst all the various disciplines that we use, and a very large manufacturing group and facilities. And so there are a lot of people that really made this happen. And these are my reflections. You might get a different slant from others that were there. So human insulin was the first product in 1978. Lilly was really responsible for development, and we assembled a team at Lilly to develop the process for it for the first time ever. Lilly was provided the constructs, the plasmid structures for the A and B chains of insulin. Remember, insulin is a two polypeptide chain, an A and a B chain, with one internal disulfide, and then the chains are held together by a couple of disulfide bridges. Probably a simple molecule relative to a lot of the things we make today. And actually, the fact that it was the size it is provided some extra complexity. So it had to be made as a fusion protein, so we hooked on a glob of E. coli protein, and then there was a cyanide bromide cleavage site, and then the A chain, and the same thing for the B chain. So this makes a fairly complicated process because you have to ferment in E. coli, you then pull the materials out as inclusion bodies, you have to purify, cleave the chains off with cyanide bromide, then purify again, then do an assembly of the chains, then purify again. So it was a pretty complex process and a lot of yield losses through all the steps. Actually, I was talking a little bit this morning and ran into the Lilly CEO, and they still use the cyanide bromide cleavage. So after all this time, you think they would have come up with something better. But part of the problem was if you don't make a big enough protein, then the enzymes just chew it up inside the cell. So that was the hand we were dealt with. The timeline to produce the product, as in all drug development activities, you want to beat the competition, get your product on the market. So it was very aggressive. So we had, clinical trials we had to do. We had a novel process and novel plant design. Usually those three combinations are a problem because we're trying to do three very difficult things all at once. There were also restrictions on experiments we could do. So the scientists, there was a famous meeting at Asilomar in the early 70s after Boyer and Cohen realized you could do some of this stuff, and the scientists were concerned and met to talk about what guidelines they would have, because no one knew whether modifying these organisms would actually be harmful. There was a lot of theory that it would or wouldn't, but nobody really knew. So they decided, okay, we can't imagine doing fermentations at greater than 10 liters, so we'll set a limit at 10 liters, and that's what they did. So and these were voluntary guidelines that the NIH enforced, but that's great for researchers. It wasn't too good for me and for people trying to develop the process, because we needed a little more quantity than that. And so there was a whole process to get equipment that you had established, set up with biocontainment, and then go validate that and take that to NIH and get approval to do these experiments. And we did that I don't know how many times. We even built a separate operation in one of Lilly's plants in England in case the US shut it down. We would have an alternate to do those experiments. The other thing that's interesting with new technology, there were some people at Lilly, Irv Johnson is one who will appear in the history of biotech and was down there for a long time, who really advocated new technology and doing things a new way, but most of the company didn't. And so you're fighting this institutional momentum a lot to get the best people to really work on the project, as opposed to the several down the line. The process is very complex. And the other question we had worked at Genentech, or they had, on a pro-insulin version of the construction. So pro-insulin is A and B chains together with a C peptide in the middle, and this is the way it's naturally synthesized. It was still a fusion protein, but you cut out a lot of the steps if you could do that. So the question was, do we wait for that and then put that in place, or do we go with the chains? We didn't. We went with the chains. And less than 5 years from the start of the project, it was approved. The single chain process was used. And it's probably largely the same except pro-insulin as it was in the 80s. And Lilly continued to be the dominant producer of insulin, although they've been overtaken in the last few years by Novo. And I think it's largely around delivery, pens and things that Novo has that have been more aggressive than Lilly. So now we're at Genentech. That's Lilly's product. That's great. They've learned a lot. They've done it, but they haven't shared a lot of it with us. We need a product of our own. And so the product that we picked then was human growth hormone. Growth hormone is also a fairly simple molecule, but a little more complicated than insulin. But its structure is very well known, or was at the time. It was used primarily for children with growth hormone deficiency. Growth hormone is made in the pituitary gland, and you could, if you got insufficient amount of growth hormone, you were really very short. And this was not only a health problem, but a psychological problem for a lot of these kids. And so the way that was dealt with was the growth hormone was extracted from human cadavers. Unfortunately, the animal growth hormones didn't work. You know, the body developed immune reactions, so you couldn't use those. You only could use human. And there was a very limited supply because of the way it had to be extracted. So we thought, hey, this is a pretty good product. We can make it the same way we're making insulin with recombinant technology. It's an unmet need, and in those days there was just the beginning of the orphan drug law, so it was an encouragement by Congress and FDA to have companies develop drugs with populations under 200,000. You got market exclusivity for that. So this seemed like a perfect project for us. But there were a few problems as we started to do this on our own. First, the analytical methods that were available then were pretty rudimentary. You know, for the protein biochemists, you know, Kamasi stain gels, where on those, product looked very pure. You know, we could purify it, it was 100% pure, but as it turned out, they weren't good enough to really use for a human protein. We didn't know how to make a protein that didn't have a start codon and then produced an extra methionine on the synthesis, and so we had an extra molecule on there. That actually turned out not to be a problem therapeutically, but it was always a worry on FDA's part. We also found out, we hired a regulatory person, we found out, oh, you need a license from the state of California to produce products in California in addition to FDA. Gee, I didn't know that. Well, what do you need to do that? Well, you have to have a pharmacist in charge of production. We said, okay, well, we have 100 PhDs, we don't have any pharmacists. So anyway, to make a long story short, there was an exemption you could take. We sent 10 people down to take the test. We got licensed, including me, and so now we were free of California FDA. We also did another interesting thing to save money, I guess. We tested the product on our own employees. Also something you would never ever do again. And what we found was the standard test for pyrogens that come from the bacterial source of the product is a rabbit test. So you inject it in rabbits and you see if their temperature rises. Growth hormone, unfortunately, suppresses that reaction. So the rabbit tests were fine, but when you get into people, everybody had a reaction. And so that required a restart. And what that led to, and the lesson for me is, you need really good analytical technology, and you need to keep that going in order to guide the purification activities in your recombinant products. And you know, that really is a must. And so a lot of work was done to develop HPLC and better gels to look at E. coli proteins, et cetera. And product got repurified, back in the clinic, was approved in 1985. And as it turns out, it did a lot better than we expected it to do, because there were a number of cases. You've all heard of mad cow disease, I'm sure. This is a disease that occurs in several species that is caused by a substance called the prion. Stan Prusner at UCSF got a Nobel Prize for this, for figuring this out. But this was the first product that was pulled off the market for Creutzfeldt-Jakob disease, which is the human version of mad cow disease, because patients developed these brain infections and died. And so they pulled the human pituitary-produced product off the market. We were there, you know, and so it became a very good product for Genentech. And then we continued to develop that. We figured out how to get rid of the extra methionine by secreting growth hormone into the periplasm. That then led to Nucleotide, which was approved in '93. So those were Genentech's foundation products. We also built our first manufacturing facility in the early '80s. And it's a little bit like here, there was a lot of thought given to proximity of process development people with the manufacturing people. They're right across the hall. The laboratories were adjacent, because we knew we were developing a lot of new technology, and we had to make that interaction a very positive one. No throwing stuff over the wall, but a lot of working together. So unfortunately, we designed it as a bacterial plant. We didn't know any better. We thought that's the way all the products would come. And that isn't what happened. One of our scientists ran into a Belgian investigator at a cardiovascular meeting, Desiree Callen, and he had been working for years on isolating a substance called tissue plasminogen activator. And TPA was, and he would work and work and work, and he'd get a microgram of it, and then he'd work and get some, and test an animal, and he'd do some more. And so he couldn't make enough of the substance to really explore its uses. We invited him to South San Francisco, signed him up on the spot, and now we had a project on TPA. And TPA is an enzyme that's involved in the anti-clotting cascade. So if you have a clot, the clot will form, and then there's a system, including TPA, to ultimately dissolve that clot. Hey, wouldn't this work on things like stroke and heart attack? If you could give it systemically, then potentially you could have patients cured of their heart attack without undergoing surgery. But certainly on the emergency setting, you could do that. And then ultimately get angioplasty or whatever, or cardiac bypass. So this was going to be our billion-dollar product. And unfortunately, if you look at the structure of TPA, it's a much bigger molecule than anything we've made in the past, lots of glycosylation sites, and so very hard to make in bacteria. We tried, and we really tried to do it. But we would get it expressed, but it was not active and kind of an amorphous mass and insoluble. One of the scientists had developed technology around expression in CHO cells, but it was really a research technique. We hadn't really done anything for a product. But we said, and I remember this to this day, we used to make money every quarter, no matter what, by licensing our products and then allowing the benchmarks to come in so we could pay for the work we were doing. We didn't want to consume too much cash. So we had $1 million benchmark with our Japanese partner, but we had to certify that we had a cell line that would make TPA. And so we said, oh, well, if we don't certify the CHO cell line, we're not going to get the $1 million. We have no idea how to make this as a product, but we'll go ahead and certify the cell line. So we did. And then they said to me, well, now you've got to go figure out how to make TPA from CHO cells. At that point, there were no continuous cell lines used for any drug product. These are CHO. This particular Chinese hamster ovary cell line was immortal. You know, it was designed to be an indefinite lifespan cell. It's non-human. That's a good thing. But there were no products approved with any technology like this, and so the whole effort became, how do we assemble a team that can do this? Because our bacterial people aren't going to help. What do we do to develop as a product? And I'll fast forward to a lot of products that are developed in the industry now. A lot of them stem from this work because the people have disseminated, but the technology really started here with the development of TPA. The first set of questions you have is there are endogenous viruses in this CHO cell. They're not functional, but they're in there. You can find them by EM. And it's possible or theoretical that other viruses could infect these cells during the production process. How do I assure they're not in the final product? Another issue that we dealt with was Desiree thought the dose was 1 milligram. It turned out to be 150 milligrams. So you can imagine if I'm making CHO product in roller bottles somewhere, and now I have 150 times that to deal with, that's not a good technology to make the product. And there were a lot of companies in those 80 time periods that were looking at perfusion reactors, looking at, there was even one company in the Bay Area using the lymph from cows where they cycle that through a bioreactor. There's all kinds of crazy approaches to this. I'll give him credit. Jim Schwartz, who was a chemie from Lilly, MIT graduate, but now is a professor at Stanford, said as we got people to do this, he said, well, look, you got this 10,000 liter bioreactor sitting here that you got for E. coli, you haven't used, let's just try it. So the cell biologist said it won't work, the cells will shear, there's no way to do this. So we tried it, and actually it worked, and that size bioreactor became the standard for the industry for a long time. And it was just because we happened to have one. So, that led to basically a new plant to make TPA. We had a partner in Germany, in Biberach, Germany, Boehringer Ingelheim, who had licensed the European rights. They built an organical plant. We had lots of virus removal steps designed into the process to be sure that it was safe, and we could convince the FDA that 10 to the minus 12 viral particles were removed by a number of steps. And we were ready, and then we thought the market was going to be so big, we kept working on the technology to develop, autocrine cell lines. We optimized media, optimized feeding, a lot of technologies that were developed, and unfortunately the market didn't participate. I think the most they ever sold in TPA was about 300 million, but it served as a technology platform for this market. Another product, 5 years from the start to approval, whole company working on it, and again, the plant and what we learned got used for many other things in the future. And Roche did their initial investment partly because of the disappointment in sales of Activase. We needed more money, so we got them to invest in 1990. And they were very good for the first, 5 to 10 years of their collaboration. They left Genentech pretty much alone to do what it wanted. The next product that we developed was human DNA. So if you're a cystic fibrosis patient, you get lots of infections, you can't process fluids, and those infections spill their DNA into the lungs of CF patients, and that's highly viscous and very difficult for CF patients to expel. So one of our scientists, who was a pulmonologist who was taking care of these kids when he was in practice, said, okay, let's make DNase. We'll give it by aerosol to these CF patients. That will liquefy a lot of these DNA secretions and allow the secretions to be expelled. Kind of a simple idea, and you needed human DNA because the animal ones weren't going to work. So that was a product that took advantage of all the CHO technology we developed and the plant. It was also a fast-track product approved by the FDA in less than 5 years, and really got another Genentech product onto the market. And that product sold a couple $100 million. And so this is a part of the reason to be in this business for all of you. I remember taking CF docs through the plant that we were building to purify DNase just as it got prior to its approval. And they're all crying. I said, why are you crying? I said, we've never had a product for this disease, in 30 years. And they take, they identify the kids when they're first diagnosed and go till about 30, and then they die. And so this was quite a gratifying thing. So that product got approved. There were several more approved for CF. Obviously, the genetic determinants are now known, so much better activity. One of the things that happened to us, I won't dwell on this, right before approval, is that cell culture, as I said, can be susceptible to infection if you're not very careful in how you operate. And this actually happened to virtually every company making mammalian cell products. And you saw Genzyme actually get acquired not long ago by Sanofi, in part because they had an infection in their plant and had to shut down. And so it happened to us at Genentech in 92, 93, probably brought in by raw materials. It's a very rare contaminant called minute virus in mice, MVM. I had actually go to the library and find an old textbook to look it up, and found one expert in the world that was still working on it. But we did develop methods to detect it, and we did do a lot of things to clean up and ultimately solve that. But it is something you have to watch out in all these production operations with mammalian cell lines. So to fast forward a little bit further, now we've got basic mammalian cell technology, and we were having debates in Genentech, are all the proteins that you could think of gone? Have you done all the ones that are likely? And really on came monoclonal antibodies. So there'd been a series of monoclonals developed that were of murine origin. They'd all failed in the clinic because of immune responses on the part of the patients. So they kind of died away. Now we've got several things that are going for us. We've developed we did develop technology that allowed humanization. So you could take the determinants in these marine-developed monoclonals and replace them with human sequences. And you could do that pretty readily, and there are a number of techniques to do that. You could also make them at large scale. Couldn't do that before. All the technology developed around TPA and DNase is now applicable to make, because the doses of these now are in grams, not in micrograms. So you have to have the capability to do that. That came along, and we had a lot of experience doing that. And the last thing, and probably the most important thing, is targets got identified in biology to be able to look at these things. So I'll talk about HER2 in a second, but HER2, CD20, VEGF were all biological targets that became quite interesting if we could develop an effector molecule to go after them. But this also meant, from a management point of view, if you remember, up to this point, we're kind of doing one project at a time. Now we had to do like 6 or 8 projects at a time. And we had to learn to manage that, and we had to learn to divide up into project teams and provide the right emphasis and decision-making in a company of, still a fairly small company, 500-600 people. So that's what went on in the 90s. We were incredibly lucky with the products that we chose and their ultimate clinical activity. Herceptin, which is still a bit the poster child for personalized medicine, was brought to us by Denny Slayman, who was an oncologist at UCLA, still is. And we had cloned the receptor for Herceptin, HER2/neu, in the 80s, but didn't know what to do with it, so they patented it and then went on to other things. Denny came and he said to us, hey, look, I've been measuring women with this receptor. The ones with a lot of copies of HER2/neu have the poorest prognosis. And if you have low copies of HER2, you do much, much better. You guys should develop, something an antibody or something to downregulate that receptor. And that was really the genesis of the project. So it came from outside. We had done the initial receptor work, but the idea for the drug came from inside. And in those days, we weren't an oncology company, cardiovascular and growth and development. So getting over the momentum of trying something new, going into a field we hadn't really been in before, those were all issues that had to be dealt with. And it turned out this was a product where first you diagnose the patient, you see if they have a lot of expression of HER2/neu, and then you treat them. And that's the way the clinical trials were done, and that's the way the product got developed. It turns out about 25% of women with breast cancer have this target, and in enough quantity where that's something that you need to do. So that product was developed. Another product called Rituxan in collaboration with Biotech was developed, which goes after CD20. So basically, its use is in non-Hodgkin's B-cell lymphoma and other lymphomas. Avastin, which is a monoclonal antibody against VEGF. So VEGF is the substance, or one of the substances that allows cancer cells to vascularize. It promotes blood vessel growth. We had a lot of ideas of how to use VEGF and anti-VEGF, but the anti-VEGF Avastin product worked the best because you could treat solid tumors, stop their growth, treat them with a chemotherapy agent, and hit them that way. Xolair was a monoclonal for asthma and allergic rhinitis. Raptiva for psoriasis. A lot of these are immune products. A few things didn't work. And Lucentis is for macular degeneration, also anti-VEGF, but a slightly different version of the molecule. So you go through all this, we're developing all these products. Now suddenly 6 of 8 products work, and we get them approved. 75% hit rate is phenomenal for our industry. Normally, biotech is about 25% of all the things you start working on and put in the clinic actually come out at the other end. Pharma is much lower because a lot of small molecule drugs drop out much earlier from tox. Probably 10%. So this was a credible lucky streak. Maybe partly we were good, partly we were lucky, but really propelled a lot of what the company value has built since. And now Genentech is an oncology company, but a lot of it stems from Denny coming and saying, "Hey, you guys should think about this," and ultimately getting us to do it. Just a last quick thing is we quickly ran out of manufacturing capacity, so we decided it was time to consider another site. All of our eggs were in one basket in South San Francisco, earthquake zone. We had a big earthquake there actually in '89, And so it was time to consider another site. I didn't want to go too far because we were still technically supporting a lot of these products, and didn't want to really move that far out. And also there's a big capacity planning question. Well, how much do I really build? You know, what if 25% are successful, I don't build for 100%. So we did look at locations in and out of California, ultimately went to Vacaville, which is an area around Sacramento. California at that time gave us quite a few incentives to stay. There probably is $1 billion invested in that plant now. It's very close to I-5 if you're ever out that way north of Vacaville, big operation. I think there's a question of how we use all this capacity and what sort of sharing we do. Some companies do work with each other. I think there's another important thing that's happening. There's a lot more disposable technology that's being used. So you don't have to worry so much about clean in place and sterilization. You can just throw the disposable technology away. A lot of companies are looking at that, particularly for early stage ones. And there's a lot of emphasis now on tax strategies. So, you know, maybe I'll build my plant in Denmark because I can, you know, keep the money there and ultimately save U.S. taxes. And so we talk about our jobs being exported. A lot of it is from tax strategies. And you can't not do it. You know, it's a hard thing for companies to decide not to do. I think also what's going to happen in the field relates to biosimilars being developed. Now, many of these products in the 90s I talked about are running out of patent life, and there'll be companies trying to develop biosimilar versions of Herceptin and Avastin over the next few years. So the technology is a lot more widely distributed. The main issues are, can you make the exact same molecule? Because the process is part of the definition of the product. How do I characterize it so I can show it's the same? How much clinical work do I have to do to prove it? But that's definitely coming. I'm going to show you a few shots if you haven't been in a biotech lately, and just quickly go through that. This is Biogen IDEC's Denmark facility, which is done and going through its validation for FDA and EMEA, the Europeans. Will be operational probably in 2013. So, but it's, I think, typical of a state-of-the-art facility in biotech, independent of disposables. This is a seed train, so preparation of the inoculum for the bioreactors. We're looking at the lower portion of that. A lot of the designs now involve gray space, so all the utilities and the kind of dirty parts are separated from where the media is added and where the operators actually work. Seems to be a good design. It's been done for the last few years. This is the bottom of a large-scale bioreactor. I think the ones in Denmark are 20,000 liters, so companies have pushed beyond the 10,000 a little bit, but it's still not much larger than that. So this is the bottom. You can see the agitators and impellers and feeds and all the monitors. This is molecular filtration skid. A lot of membrane technologies are used now in processing. A lot of media is fed. Most of these reactions are run as fed batch. So you have a batch reactor and you're feeding it periodically with media to optimize the amount of product, and then you harvest as a batch. And so there we've got the buffer prep lower, buffer prep upper. You see the facilities are pretty nice. You know, a lot of shiny stainless steel, a lot of well-designed piping, and you know, we've evolved to a much better design as we've done a bunch of these kinds of plants. Chromatography is used extensively to purify all these proteins, and this is a typical chromatography skid for a large-scale production. Typically, you try to combine affinity, size exclusion, and ion exchange all together to ultimately purify the final product and get rid of the impurities that are associated with them. And then in mammalian cell processing, incorporate steps, membrane steps, or heat pH steps to get rid of any endogenous virus that might be there, although that's largely theoretical. And this is ultrafiltration, so as you get to the end of the process, you have to concentrate the product protein again to get ready for formulation and storage, and UF is used a lot for that. I'll just touch quickly on disposables. I've become kind of interested in this, and so is Biogen IDEC, as a way to be more flexible. And now there are technologies at virtually every step in purification and bioreactors. There's a gizmo that you can see the waves is a big plastic bag. Cell culture goes in that, and the thing goes kind of like this and aerates, and and then when you're done, you throw it away. So I think that technology is going to help small companies make the first quantities of their product. It may make larger companies be more flexible, help them to be more flexible, and avoid these. You can imagine the capital cost. Denmark facility was $500 million just for that first phase. So you have a lot of fixed piping and sterilization and control. So it may be that'll help with efficiencies in the industry. Okay, so are we doing okay on time? We're done, right? 5 minutes? Let me see what I could do in 5 minutes. Let me just let me take you through quickly Biogen IDEC, and I'll just highlight a couple of things there. So Biogen was one of the original biotech companies, along with Genentech, Genetic Institute, CETUS, and the like. Founded in 1978, a couple of Nobel laureates, Wally Gilbert and Phil Sharp from MIT, were founders. Their early products were competing with Genentech, a lot of them, but their biggest success came with something that was developed for molecular sclerosis. It was actually beta interferon. It's called Avonex is the brand name. And it's helped thousands and thousands of MS patients worldwide since it was introduced. And it was a little bit of a fluke. They were developing things that Genentech was developing. They decided not to do that. We weren't doing beta interferon, so they decided to do that. And I don't know who it was that decided to try it in MS, but they did. And that became their first big product. Then in another company in California, IDEC, started by an ex-Genentech scientist, just developed an antibody for non-Hodgkin's B-cell lymphoma called Rituxan. This was a cancer product. It targeted CD20 and basically wiped out your immune system, your bone marrow immune system, and then you got relief from the disease. And so that was a collaboration with Genentech. The product got approved. And then in 2003, Biogen and IDEC merged and formed one company, Biogen IDEC, headquartered in Cambridge. I was on the board of IDEC, and so I stayed on the board as we merged with Biogen. And now we have a very exciting company. We have Rituxan, which Genentech sells and is the partner. We have Avonex. We have another product for MS called Tysabri, and those are all approved. And then we just got results, and we have some long-acting factor VIII products for hemophilia. Those are in the clinic. And we just got phase 3 data for an oral, very active oral product called BG12 for MS. So the company's basically focused in neurology and some immunology, and I think 4,000 employees, $5 billion in sales, geographical footprint, worldwide footprint, new CEO who's doing a great job, and so I'm very excited about all of that. There, in fact, on the right is the facility in Denmark that I showed you that's undergoing its validation, and Zug is the corporate headquarters. BioMarin, I think I will skip. Theravance, I think we'll step. Let me talk a little bit about personalized medicine, because it hasn't come as quickly as I thought it would back when I went to my last company, but I still think this is an obvious and great way to make drugs more efficient, and one of the few cost-saving ideas there are in healthcare. So some of this comes from the fact that we understand biological processes better, and that's going to lead to new targeted drugs and has led to new targeted drugs. Unfortunately, many of the breakthrough drugs don't work in all patients, and if you look at response rates in oncology, they're 20 to 50%. We don't know how to use them in combination. We don't know which patients are candidates. Herceptin is a great example that's different. That one, we started with a diagnostic, but most of the great cancer products, we have very little idea of how to use those. But molecular diagnostics, I think as we learn more about pathways, will have a significant impact on this whole field, particularly in new and developing products that aren't yet on the market. And I was explaining this to some people the other day. If you think about, I've already told you about the Herceptin example. You think about the treatment of HIV. So here's a disease that not too many years ago was a death sentence for anybody who was infected. and basically you could take care of the patients and deal with the side effects, but there was nothing really you could do. Now we started developing antiviral drugs, and those drugs were very powerful, but the one disadvantage they had was virus, being as clever as it is and very easily mutated, would quickly escape the surveillance of those antiviral drugs. Okay, then we said, let's use those drugs in combination. Same thing, a lot of resistance developed. And so my last company and several others developed technology to be able to look at that patient's HIV by various molecular biology techniques would not be possible a few years ago. Understand which drugs were failing in the patient's regimen and which alternate drugs were options for that patient. And it often was not all of the alternate drugs that he wasn't on because there are class effects. So a given protease inhibitor might also, the second one might also be a poor choice. But anyway, that technology got used to manage the disease. There are now 26 drugs approved to treat HIV. Patients are going longer and longer without failure. We can't cure it yet. Maybe we won't for a long time. But we can certainly manage it as a chronic disease, and that's basically what's happened. You can think about the diagnostic tools as a big part of why that's happened. And there's no reason we can't do something similar in cancer and in other fields as the technologies develop. And again, I think HIV is an often overlooked but really important example. Now, a few years ago, the drug developers were pretty ugly toward personalized medicine. J.P. Garnier, who was the last chairman of Glaxo, was quoted as saying, "Don't mess around with my drugs, mess around with somebody else's," meaning that he didn't want to have the drug on the market and then be told that the market was half of the size that he had anticipated. But I think that's changing, and I think now in most companies, biotech and drug, there's much more interest in finding biomarkers to go along with the drug development program. And this is a quote from Monday from Hal Barron, who I know well, who's now the chief medical officer at Roche, who was at Genentech, and basically saying, look, the probability of success of a successful clinical trial can be increased dramatically by, I would substitute molecular diagnostics, but it can be genetic information. And you're going to see a lot more examples of companion diagnostics as we go forward. I think this is all really important trend, and I think that it's going to happen and has happened increasingly to additional products. I'm not going to touch on NanoString. We'll save that for another time. But so here are some challenges. Rex and I were talking about this. So personalized medicine, that sounds like a great idea, Bill, but what are the barriers? Well, we have a few. Some of it is technology. Okay, if I'm having a molecular diagnostic, do I look at the DNA? Do I look at the RNA? Do I look at proteins? Do I use mass spec? There are so many possible ways to approach this that sorting all that out is going to take some time. Second, there's a lot of data, but not much of that translates into real information. And by real information, I mean, well, what does all the sequence data mean, and how does it translate to clinical outcome? That's what you really want to know. And so that's going to take time to develop. You're going to need clinical data. You need to make these measurements and then correlate it back to how patients do. There are two regulatory bodies that deal with diagnostics. One is the FDA. So if I have a kit and I want to sell it across state lines, I have to get FDA approval, and they're increasingly requiring more and more clinical data. If I want to keep it in a service lab format and just do it in California, I can do it under something called CLIA, which is administered by CMS, and that's a whole different set of regulations. And so these two regulatory things kind of compete, and that's an issue that needs to get resolved, and at least in 10 years has not. It's still kind of a back and forth. So there's some very important companies that have given up on FDA and said, I'm going to do the CLIA model. Genomic Health in California is one of those, and they have a 20-gene signature where they can predict patients' response to chemotherapy. You should or shouldn't get it when you're first diagnosed in breast cancer, and it's a very successful company. So they've stuck with the CLIA model. Other people do the FDA model. I think that, as I say, it's information, so access to clinical specimens and that clinical trial data, usually always in the hands of either investigators or the drug companies, is key to getting this done, and cracking that and getting everybody to open the data and be able to use it more successfully is important. The drug developers want to control what's going on the diagnostic side, so there's always that tension. The reimbursement side is pretty funky in diagnostics. They've set up a system where you only get, if you use the formal system, paid for the work you do to run the assays doesn't make a lot of sense. If you create value, you should get paid for that value, not for how many steps you have to do to create the assay. So a lot of debate about how to change that system. And of course, overlaid in all this is all the healthcare cost pressures that we have on us. Regulatory confusion, that comes from what FDA is or isn't going to do. And I think healthcare reform should care a lot about this, but there's nothing specific in the healthcare reform law that would promote or even incent personalized medicine. So, but I think all this stuff's going to get addressed, and it started to. There are more products this way, and I'm very optimistic that we're going to have very efficient drug development, both in the clinic, because we can save money by treating fewer patients and having more certainty of efficacy and safety, and also ultimately commercially. So, in summary, Biotech's alive and well and doing really good. I think I was listening to a talk by the president of Rockefeller the other night, and he actually is a former Genentech scientist, and he said, hey, in biology, we're at the beginning, we're not at the end. We know a lot more about how these pathways work. There's a lot more future for innovation. We'll do it in a much more intelligent way. And, you know, the bottom line here in our industry is not only can you do well, but you can also help a lot of people, and that's what we're here for. So anyway, hope you get into the industry as you get out of chemical engineering. Glad to help any of you if I can, and I wish you the best. Thanks very much. I do. I have to go home to California. I'm really sorry.
But we have about 10 minutes, so let's start with the two first. You only have two.
I would, yeah.
All right.
Just out of curiosity, I was wondering if you could comment on sort of the early slides that you're talking about, how much more expensive it gets to develop drugs. I was wondering if you could maybe break down a little bit how much those expenses might be coming due to regulation, like from the FDA, as well as due to sort of, to put it poorly, the industry having picked all the low-hanging fruit, let's say, and now making it more complex.
That's a great question. I think it's a little of both. I think that we were in an era where a lot of the easier targets did get picked, and now we're in harder targets, and so there are more failures. I think what you aspire to do as a drug developer in biotech or in pharma is you want the failures to happen earlier, in phase 1 and phase 2, and in the preclinical work, not in phase 3. About 80% of the development cost is actually in phase 3, and the increase in those costs, a lot of it is driven by FDA wanting more data. So there's a little bit of both in there, and you could imagine, if you want to be an optimist, that going forward we're going to get a lot better because we're going to be able to pick our targets better. There's more knowledge about the biology of disease, and that should help. But biology is a tricky thing. If you interdict at one place, there's some other place over here that come back to get you. But I think it's a little of both. FDA is the most conservative I've ever seen. And it really is a problem. I think it'll swing the other way, but there's an equation here. If I have a drug that would save 100 people, and I might hurt 1 out of 100, should that drug get approved? Okay, so I avoid hurting the 1 out of 100, but I also avoid helping the 100, and that's always the equation that you have to deal with. No drug is 100% safe, and so FDA is over here protecting the 1 out of 100, not so much the people who need the drugs, and I think that's a problem. Yeah.
So if you will just say that one, it's not a problem. So what would be there to benefit FDA?
They'll never tell you, but the desire is more and more data to show that whatever safety problem signal you see is not a permanent one. I think one way to deal with it is to say, because you can't possibly see all the side effects of a drug when you do clinical trials because the patient population is too small. So one way to deal with it is to say, okay, do this study to get approval. Now I'll do post-marketing studies to look at safety, which will be more patients over a longer period, and I'll do that, and I'll commit to doing that. That would be one way to deal with it, but they so far haven't done that. They're actually doing both. They're requiring more data and the surveillance. So. Okay, I guess everybody's going to oil companies, no?
You mentioned one case about this,
Yeah.
You mentioned in the one case was the initial one, that the process is ugly and it has been the same for 30 years. Now, this seems somewhat difficult to understand. Why is it that it's under the out for 30 years, but somehow, can this be, I don't mean in terms of making it elegant, but I think of making it better somehow.
I agree with you. I was surprised to find that out when I ran into the CEO of Lilly a few weeks ago, and now he didn't give me all the details. I'm sure things are a lot more automated. They know more about the process, but its basic structure is the same. Yeah, you would think that in all this time it would be. I think there's conservatism in a lot of biotech processing that I think we're on the verge of having disappear. Mainly because of the pressures and costs. So if you think about, I spend, $500 million to get my drug approved, including all the manufacturing I have to do. I have to build a plant. I put all that together. In biology, in biological products, the process is actually part of the definition of the product. Now I'm going to improve it. And I look at that and I say, okay, what are the risks of improving it versus keeping it just like it is, and all the regulatory issues I'm going to have if I try to change it. And I think that's what a lot of companies face, and they end up deciding in the more conservative way. So if they decide to build a new plant and they're going to go through the license again, then they can import new technology. But when I went to, I'll never will forget this, because a lot of the IDEC scientists were from Genentech, so we were very aggressive about process improvement and yield improvement. And so we merged the two companies, IDEC and Biogen, and then we look at the Biogen legacy processes, and they were terrible. I mean, they hadn't done anything in 20 years. And but I think a lot of it is from conservatism on what you'd have to deal with on the regulatory side. With biosimilars coming and more spread of technology to other countries and other places, and the need for more efficient processes, I think that has to change. And there's a lot of things that can be done.
I was wondering, in your experience, what happens culture-wise and I guess company-wise when a company transitions, especially in the healthcare drug industry market, when it transitions from a privately held venture capital through the IPO and into is there a culture change?
Yeah, that's a good question. I mean, certainly there is for the CEO and for the top management, because you now have to spend a lot of your time satisfying the public markets that are your investors, and it's comparatively easy to do when you're private because you know who they are. There's a handful of them. A lot of them are venture capitalists or early stage investors. So yeah, it's very different, and does, I think, take away from the top management's attention to the business, because they're busy doing that all the time. This is a philosophical question, but I think the model of having a public company with today's investors that develops drugs and the timelines in drug development is sort of incompatible. Because investors want returns every quarter. They want to see the money go up. They want so that's their focus. They don't care what's in your pipeline 5 years from now. And yet the company, to be sustainable, needs to continue to develop new products and acquire them some way. And those models are right now not compatible. That's part of the reason the IPO market's not working, and part of the reason that pharma companies are under so much pressure. And in fact, a lot of investors will tell you that they view the R&D dollars that are being spent by pharma as wasted, and they'd rather have the money. You can give the money by dividends, or you can give the money by buying back stock. That's essentially the same thing, because the number of shares drops and the shares value goes up. But that's the climate for a lot of investors today, and that's not a good thing. Not a good thing. Because we're in a long-term business. We're there to develop drugs. Technology takes time, and if you're held to this quarterly thing, it detracts from that a lot. So I don't know what the answer is to that. Probably can't all be private, but it may be that investors need to take a slightly different view if they want to invest in healthcare, that it's going to have a longer cycle. And it used to be that way. You used to have a lot of retail investors. I remember when Genentech was acquired by Roche, we had these annual meetings. Nobody comes to annual meetings anymore, but then you had this packed room, much bigger than this, packed with people. A lot of them mom and pop investors. And one guy got up and said, and we were buying back, Roche was buying all the shares from the market, and one guy got up and said, he said, "You're betraying me." I said, "Well, why is that?" He said, "I got on the train when you went public, and now you're throwing me off." So a lot of investors had very long-term views. It's not so much that way now.
Jim, two questions I've seen now. Are there some questions? Okay, sir.
Just interested, following up on the conservatism one. In small molecule pharmaceutical processing, in the farm development, and the Dutch solid state forms, the pharmaceutical industry is still very conservative, even on those relatively straightforward processes. But it's changed over time. The industry only started getting lots of engineers involved in that stage of the process relatively recently, and it seems to me the companies that have more engineers in them have become more aggressive on process development, because chemical engineers, that's where they get their kicks out of improving processes. Is there an analogy in the biotech industry? I mean, the different companies have, some companies run by biologists and organic chemists, and some have more engineers, or?
I think probably so, and that may explain the Biogen versus Genentech philosophies. We had engineers employed from the very beginning, maybe because I was there, I guess. But we had, it's very multidisciplinary. So much like you do today on the campus, where you interact with the biology groups and biochemistry, we had to do the same thing. So we had mixtures, but. My first consultants were MIT engineers, and in those days, Danny Wang and Charlie Cooney were the kind of biotech guys at MIT, and they worked with us a lot, and so yeah, it was a mixture. I didn't see that so much at some of the other companies. So you may be right. It may there may be different philosophies, yet. It turned out that you needed all those skills to really make this stuff work, because the biologists, they brought their part, but they couldn't do it all. So engineering is very important.
Just one word to what you said. So I see that we mentioned that for 5-6 years they were learning some from Cooney.
Yeah.
But after 5-6 years, it was turning out they were learning more. The consultants were learning more from them, but they had gone so far ahead, so they had not to do things on their own.
No, we did, and the lesson for me was I went around to get people. Charlie would come and say, and I love Charlie, he's a great guy, but I'd go around and say, to my top people, "Hey, can you talk to Charlie? Do I have to talk to him again?" So it was at the point where we weren't learning much from him, and it was mostly the other way. So.
We will take one last one before 5 minutes after running on this. One last one.
Yeah, for 100 years, this place is known as the School of Chemical Engineering. I was just wondering, looking ahead for the next century, should we consider adding biomolecular engineering into our game? I want to take this up.
In my opinion, yes, but I don't know what the rest of the faculty thinks. I mean, this area is so important that when I went through here, we were all trained for chemical companies or oil companies, and those were the job offers you got. It was really only a fluke I ended up going to Lilly, and I learned a lot there. But I think you can do, if you don't call it that, you should have a strong curricula in biochemical engineering to go along with the Chem-E work, and let people choose after they get the basics. It's really important, and you know, to give alternatives to students who want to go in different career directions. And I think Biotech's going to be here for a long time. So.
Well, Bill, thank you very much for all your remarks and your time.
Yep. There's more stuff. Oh, thank you. Yeah. Oh, okay. Oh, thank you. Thanks very much. So then it's been great. It's been great. And if I can help, you know, in the future, let me know. And,